Hello, my name is Lorraine Foley and I am the PI of Leaf No Waste. This research team is a collaboration between TU Dublin and Teagasc, and we are funded by the SFI Future Innovator Prize.
Hi, I'm Lael Walsh and I'm a Research Officer in Sustainable Horticulture at Teagasc.
And I'm Lorraine Foley, a lecturer in horticulture at Technological University of Dublin.
We'd like to talk to you today about our research in spinach production and how we're trying to reduce food and plastic waste. We're trying to make spinach production more environmentally sustainable.
Environmental sustainability means thinking about the impact on the environment of the growing, processing and packaging of spinach. We're also looking to reduce the waste that occurs when consumers don't use all of the spinach they buy. The situation of food waste in Ireland is really quite scary. We waste about 1 million tonnes of food every year. As a nation, we're responsible for an average of 54 kilos of plastic waste per person every year. That's the weight of 54 bags of sugar.
We use packaging to keep the environment of leafy veg constant, and the right packaging can reduce waste of perishable foods like spinach by keeping them edible for longer. But this packaging is often plastic-based, and synthetic plastics negatively affect the environment. So is it possible to reduce our reliance on plastic packaging and the risk of food waste?
Well, this is what we're trying to find out with our research Leaf No Waste project. This is a project that's supported by the Science Foundation of Ireland.
What we know about spinach is that it continues to breathe after it's harvested. This is known as respiration, and this leads to moisture buildup which can cause mold, fungal, and bacterial growth. And this causes the spinach to spoil. We want to lessen this breathing through the use of biostimulants, or as we call them bio-based products, applied to the plant while it grows. This tackles food waste, but what about plastics?
There are some better packaging types that are biodegradable, compostable, or which can be recycled. We need to make these types of packaging more suited for use with spinach. If we can make the crop last longer and ensure it's packaged in a more environmentally sustainable way, this will lower the impacts on our environment, reducing food waste and global warming.
I'd like to talk a little about the results we gathered during the experiments of this year. During the seed phase, we built upon our knowledge from the concept phase and increased our range of crops. Leafy green crops such as spinach, mustard and kale were grown from seed. Silicon was applied as a foliar spray in 3 concentrations twice during the growth. At maturity, they were harvested and transported to the Greenway Hub in TU Dublin to be packaged in OPP and PLA plastics, whereupon extensive post-harvest testing was conducted. Post-harvest testing in OPP and alternative compostable plastics shows silicon has an effect on quality. Weight loss is notably greater in the control samples compared to all the silicon treatment samples. In Figure 2, the chlorophyll levels are reduced in the control samples in comparison to the treatment samples. This may suggest that silicon encourages the development of more chlorophyll during growth. Here we have data that depicts comparisons of gas compositions within packaged fresh produce, kale and spinach, on day 7. To avoid food spoilage, gas composition for packaged produce must not exceed 10% carbon dioxide and 2% oxygen has critical limits. These critical limits are illustrated as red lines in each of these charts. Our experimental data provided the framework for this simulation that describes the frequency of degraded produce under different treatments. The treatments are listed along the top of the chart, and the packaging solution, produce and gas type is listed along the right-hand side. If you observe the spinach control for CO2, you will see that the frequency of produce exceeding the 10% threshold is slightly greater than the treated versions. This pattern is repeated with the O2 readings on the lower part of this chart. The control for spinach, for example, there are some samples that exceed a 2% limit. However, the treated materials have no rise in oxygen. Reduced respiration rates are linked to longer shelf life for fresh produce in sealed packaging. These results suggest silicon treatments reduces the risk of spoilage. This approach allows predicting the probability of food waste occurring for a range of treated produce with consideration to the produce variability. We collected data from conversations with stakeholders and crop trial experiments. We completed the first life cycle analysis for spinach and created tools to measure probability of food waste. Next, we will increase our portfolio of commodities to include soft fruit and mushrooms. Factoring in results from consumer surveys and studies and sustainable opportunities will help build our green business plan. We wish to align our goals with that of the EU Farm to Fork strategy.